microscope, essential input from mouse cell types, from MIT, from neural coding. It has a really wonderful, nice group of scientists, and it's looking at one of the most important questions in neurobiology, the morphology of single neurons, and really doing it in a beautiful way. And it's a great group that ranges from the far northwest corner of Russia to a resort town in Turkey, to Lisbon, to Lubbock, Texas, to Seattle, where they're all from. And it has a group of names, at least 3 of which are hard to pronounce, and we all do a pretty bad job of them, so I'm going to be very careful. Anton Arkhipov, Ali Chetin, Stacy Sorensen, Shomo Chatterjee, and Nunu Da Costa. Please.
Thanks, Clay, for that introduction. So I'm Stacy Sorensen, and I'm happy to be the first speaker here from the Allen Institute today. And in this series of team talks, we're going to move through multiple approaches to characterizing the structure of the cortex. So I'm told this thing works. Oh, there we go. So I'll start things off by talking about the morphology of single neurons labeled in mouse visual cortex in vivo. And the data that we generate for this project should be very helpful in helping us to answer some of the questions that we were really interested in here at the Allen Institute, such as, oh gosh, sorry, I got to figure out the difference between the laser pointer and the, oh my gosh, I don't even know how I ended up. This is the end of the talk. Okay, got it. Left, right. Oh my goodness. There goes my 5 minutes. So we're interested in the properties of a neuron that can define a cell type. In particular, since we're focusing on morphology, this is my laser pointer. There we go. Do the dendrites, does the branching pattern of the dendrites shown here in red, the trajectory of the axons shown here in blue, do those things matter for understanding what's unique about this particular cell here? And then based on these characteristics, as well as a number of others that people will talk about today, how many cell types are there? We're starting in V1 and we'll move on from there. And then how do they connect to one another? So this is just a simple circuit diagram that Nuno put together that's supposed to represent the potential connectivity within mouse V1 or any cortical circuit for that matter. Okay. Yes. Okay, so I thought I'd show you a video of the adult connectivity atlas, just to give you a perspective on how we do this project. Basically, for the mouse connectivity atlas, they inject AAV virus into the brain and then trace the projections between regions. And then what we're looking at here is in the greenish yellow is the projections of about 10,000 neurons sampled at every 100 microns. So for the current project, for the methods are actually quite similar, except for instead of labeling 10,000 neurons, we're obviously labeling 1 or 2 neurons using DNA electroporation in vivo, and then instead of sampling at every 100 microns, we're sampling it at less than 1 micron. So this higher resolution imaging has the potential to generate much larger data sets, but we get much more detail so that we're able to see things like individual spines and follow the trajectory of individual axons locally as well as in the long range. So with the connectivity atlas, it's extremely beautiful, but you have to deal with the tangle of thousands of axons coursing through the brain. And so here we're actually able to follow single fibers and in great detail. So one of the things that we do have to do for this project is that we need to align all the sections that we
Stacy talk about reconstructing individual neurons at a level of completeness that I don't think we've ever seen in mouse visual cortex before. And of course, this is part of a much broader push in the Institute to not only use anatomy, but biophysical properties and molecular markers to come up with a systematic way to define individual cell types uniquely. Creating a dictionary, if you will. But if someone handed us such a dictionary right now, we would still be hard-pressed to figure out how the functional identities of these neurons arise, because that is a function of the circuit in which it's embedded. The hundreds or thousands of cells that provide inputs to that one cell, and then the biophysical algorithm that the cell does and create an output, that we cannot know from this. So this is just an example of the kinds of two-photon imaging data that we get. These are all neurons filled with GCAMP6. They are firing, and from the firing we can extract tuning properties and things like that. But we don't know which neuron is connected to which, and who is providing input to whom. And without this, we can't get at how this functional identity, the creation of receptive fields, the spatial-temporal receptive fields, comes about. So we need a way of labeling a neuron and all the neurons connected to it. And right now, the only game in town that I'm aware of that can do this in vivo, without having to do in vivo, ex vivo correlations, is tracing with rabies virus. And so briefly, very, very quickly, okay, what's going on? So rabies is an obligate retrograde tracer, which means that if a neuron is infected, it goes up the dendrites, crosses the synapse, and infects the presynaptic cells. And wild-type rabies, of course, would keep doing this until your entire brain is rabid and you're hydrophobic and bad things happen. But rabies can be engineered in such a way that it infects only a single cell. It expresses something that we can see, a fluorescent protein, crosses a single synapse, and then stops, giving us the monosynaptic circuit in vivo, and we can see it, and we can also get the function of these cells, so we can understand the functional identity and how it comes about. This is just an example of a single cell labeled and all the presynaptic partners of that cell. Very quickly, what does an experiment look like? So we first image cortex. This is flavor protein imaging, basically a form of intrinsic signal imaging. We define where V1 is. Then we go in with a pipette, micropipette, high resistance. It has a contrast agent in it, so you can see, I don't know if you can see, but you can see neurons in shadow. You approach a single cell, the cell that you want to study. You touch the membrane, pass about 12 volts through it and a train, and label that cell. And you label it with plasmids that express DNA that the rabies recognizes and that the rabies needs to cross a single synapse. And this is a recently obtained presynaptic tree. Can you make the movie run? Is there a way? Thank you. So these are, within this field of view, this 400 by 400 micron field of view, these are all presynaptic neurons to the central cell of interest. And this is the anatomical part of the project. Now, of course, anatomy is nice, but we also want to get at the function of each of these cells. So how does one do that? All right. Bistable percept. Hang on. Next slide, please. So there are a number of ways, and we're pursuing two strategies right now. One is, so of course I said that the rabies can express something that we can see, and in this case we can put GCAM6, which is a calcium indicator, into rabies. Basically, the neurons that you're seeing now fire, and you can watch them fire, and you can watch how the firing influences the central cell. Another way is to make, say, for example, a red presynaptic tree and take one of our many transgenic mice, and this gives you the anatomy in the red channel and the function in the green channel of the presynaptic cells plus all the other cells. So we can see shifts in activity if a cell is connected or not. And this is basically, in conclusion, a way of bridging a very large body of imaging data that we're going to be getting from, there are pipelines and other things that we'll be getting population data and a lot of single cell data. So this is a way of actually bringing the two together. Thank you.
All right. My name is Ali Setin. I'm at the Research and Development Department under Hong Kui Seng. I will start with the analogy Alan Jones started with too. He mentioned this telephone, iPhone 6, which a lot of us are looking forward to. Imagine dissecting the iPhone 6 by just looking at the cables themselves, right? You can find a bunch of transistors interconnected. That's extremely valuable information for us, right? We would like to do the same thing in the brain of mouse, specifically in the visual cortex. So the way we do it is basically trying to dissect the circuit utilizing the Cree driver mouse lines that express the Cree recombinase in subtypes of these neurons, call them the transistors. This will give us not only the information about the axons, but also about which type of cell is connected to which other type of cell. So this is the same system that Shomo has been describing to you. You have the MV rabies that has been engineered to have this glycoprotein, which only can infect the avian species. And that way you can introduce the receptor into a mammalian cell and generate a docking site. And it's all Cree-dependent. Once you have the infection, through the complementation with the glycoprotein you have here, which makes the virus go retrograde, you can generate a monosynaptic map. So this has been developed and working, but what we did is try to optimize this system in a way that we can get less spurious labeling and more of the input supplying neurons, and generate a complete map, relatively better map of the visual cortex. Well, eventually a complete one, hopefully. So this is the experimental workflow we have. We inject the AAVs that generate these complementation system into visual cortex in multiple different Cree lines. In this case, we have the NR5A1 Cree that specifies the expression into layer 4 subtypes of excitatory neurons. And we wait for 3 weeks, and then we inject our MA rabies, and then we will see the spread after a week. This is the constructs. These are 2 constructs we generated in the lab, which seem to be working at the moment very well. And this is the MV construct that expresses the GFP. Again, reminder, this is a rabies that has been modified. It can only go retrogradely at a single synapse because it lacks the glycoprotein. And this will complement with that. Okay, so here are our cells over here in the visual cortex that are expressing the TVA, the DS-RET fusion, and you can see their presynaptic partners. And if you look at the LGN, you can see a stripe of neurons at the bottom, which are connected to these cells. So this way, we can generate a monosynaptic map of the entire visual cortex, entire visual system. So another thing we have been focusing on is trying to generate methods which will allow us to do a more high throughput style of single cell genetics. For example, these things, the types of things that Shomo has been describing to you, electroporating the DNA into individual cells. How can we bypass that step? So what I envisioned initially is to generate a system in which if you shine the light in a single neuron, it will induce a genetic mark on that neuron, and then recombine, let's say, a system of induce a system of recombination in which a gene is turned on. So what we did is we basically chose the same Cree recombinase I was describing earlier. We put it apart into 2 pieces. We attached light-sensitive domains coming from a certain fungi, and when we shine the light-sensitive domains interact with each other, and you can induce recombination, as you can see at the bottom over here. our first in vivo data from that. We have the 2 Cree components injected into brain, and in the control where we don't have light, you can see a single cell spheres recombination, but in the light-induced state, you can see lots of cells. So hopefully in the future we will be able to do Cree recombination at the single cell level in a very high throughput manner with this methodology. Thank you.
So you have seen how we investigate circuits with light. I'll discuss a bit how we do it with the electrons using the electron microscope. And one of the milestones that we are interested in is this W alpha beta connectome, which is basically a matrix in which each entry is the strength of a connection between 2 cell types, cell type alpha and cell type beta. And strengths can be measured in many ways, but the way I'm going to discuss it here is by number of synapses. And the example that I'm going to use in layer 4 of Miles' visual cortex is the input from the thalamus. So we have labeled the thalamic input with an antibody, which allows the thalamic botons to be black at the electron microscope. And basically then what we do is just count how many synapses are formed by botons that are black and how many synapses are formed by botons that are not black. And by doing that, we can get a ratio of a percentage of synapses in layer 4 of V1 that come from the thalamus. And the number that we got was quite surprising, about 30%. And it was surprising because in the cat and the monkey, where these other measures have been taken, it is much smaller. And we are now going through controls and evaluating and repeating this experiment again and again. So, but it's not the case that in all pathways you can label all the input and all the synapses that an input forms. So it would be quite nice if we could just, by looking at a button, we could say from which cell type it comes. And by doing that, we wouldn't have to do the labeling, and we could even just go through the dendrite of neurons and see their location, the location of the different inputs. And to do that, Agnes has been, again, in layer 4, checking if this approach is feasible. So she had labeled botons from different cell types coming to layer 4. On the left side, you see a layer 4 input from an NR5A mouse, labeled with gold. In the middle, see an input from layer 6, labeled with DAB from an NTSR1 mouse. And on the right, you see the thalamic input, again labeled with gold. And she had reconstructed this in 3D and take several measurements, of which here we just showed the distribution of synaptic sizes between layer 4 and layer 6. So now, based on these measurements and others, Stefan Michalis had run a classifier, or even though our data set is not that big, now it's approximately 80 to 90 botons, you can already discriminate between layer 4 and layer 6 botons with accuracy of approximately 85%, which is quite promising. So now, the other project that we are also working on, the other milestone, is imaging at the synaptic level a cubic millimeter of cortex. And this, in order for this to happen, we need technology that allows us to cut these sections and to image these sections. And for that, and if we have such a volume, we can now look at this matrix of connectivity where the axes are not cell types, but they are individual neurons with their private connectivity and their private physiology. And to do this, we are using the technology that Clay developed in Harvard, both of the sectioning by delivering the sections in a tape to the TEM, and also by adding camera arrays to our transmission electron microscopes. And you see here now on the left a picture of Derek, our EM wizard engineer from Colin's team, working on our TEMCA system, which you see there. And what you see on the right, the big picture, is one of the first pictures collected, EM pictures collected from our TEMCA system in Seattle. So now, when you collect all this data, you still have the very, very, very big and hard task of reconstructing this tissue. And either it will be reconstructed by a machine or by a human. It will be much easier if at least some of these botons are labeled black. And what you see on the bottom row, it's some pictures of a Cree-dependent expression of a EM tag called Apex2. It's a peroxidase that Ali has developed for us, and it's working. And on the first two pictures, you see it's targeting spines. On the last one, it's targeting botons. So it goes through all the different compartments. before I pass to Anton, I would just like to say that this is just a little bit of what we do in neural coding, and where we hope to be in a few years, to be able to integrate all the different knowledge that we have from physiology to EM in a kind of this circle for which the MAT team will be quite important on not only on analyzing the data, but also driving some of the questions that can be gathered from this type of data sets. And so I pass it to Anton.
Okay, so now that we heard these talks about the various beautiful experimental techniques that we are using in the Institute to characterize architecture of the brain, let me tell you a little bit about our modeling and simulation efforts, where basically we are trying to incorporate as much as possible of the experimental information collected in the Institute to develop these models to characterize brain function and activity in the brain, to create better understanding of these processes and the mechanisms underlying them. And so I don't have time to go into details about all the different modeling projects that we have here at the Institute, but let me just give you an idea about about our general workflow in all of those projects pretty much. So this slide illustrates such workflow. We start from components. So, for example, single cells or single synapses, and we combine experimental information obtained in-house. So, for example, morphology of neurons, like what Stacy told you about, and also electrophysiology, intrinsic properties of the cells. You will hear more about this later during these 2 days. So we combine this information to create models of individual cells, and then we use them as building blocks to create models of networks. And there, for the connectivity and for synaptic properties, we again use the information obtained in-house, such as that described to you by Nuno, Shomo, and Ali. And finally, we run simulations of these systems, and we compare the outcomes of those simulations with in vivo measurements, such as those described to you by Shomo, but also other types of measurements, electrophysiology, extracellular, intracellular. And so let me just illustrate you for you one such project. So this movie shows very dimly an example where we are trying to build a model of layer 4 of the primary visual cortex in the mouse. So here we start with this is preliminary work. So we start with just 2 model cells, a pyramidal excitatory cell and interneuron, and we put them together. We replicate them many times over to create a system of about 10,000 cells. So now excitatory cells are shown in red and inhibitor in blue, and we run a simulation whenever a cell fires an action potential. It's highlighted here in the movie. And now we use actual recordings by some scientists here in-house from the LGN of the mouse observing visual scenes on a computer monitor to drive this simulation, right? To supply the stimuli to this model of the layer 4 of V1. And then the activity that results in this simulation can be compared potentially with recording from actually the same mouse, but now from V1. And so by doing this many times over and going hand in hand with the experiment, we hope to discover some mechanisms underlying this sort of activity and potentially propose new experiments. And finally, I just want to point out that we work here at different levels of abstractions in terms of modeling. And please come and check out these posters where you will see more details about each of this method. But in a nutshell, we have 3 sort of levels. So biophysically detailed models where we represent morphology and biophysical properties of neurons explicitly. We have point neuron models where we represent neurons as points with certain properties approximating their behavior. And finally, population statistics models where a population of neurons is described by a single equation, and one can simulate a number of such equations to go up to a whole brain simulation at a very rough level of detail. And finally, please come and check this poster, which highlights our interactions with the talk. Thank you very much for your attention.